All right, so very good morning to all of you and welcome to APA India 2021 webinar series. Today is webinar number 15. The monthly theme is biotherapeutics development and today is a CMC session. So we will get started with a small promotional video from Thermo Scientific and then we will officially start the webinar.
Challenges scare me, I'll be honest with you.
There were a number of challenges. There were a lot of changes.
Things are changing all around us every day.
We needed to figure out how do we meet the supply and demand for our customers.
We've had a requirement to increase capacity of our facilities.
We have labs that can normally handle 20 to 25 people. All of a sudden, we're only able to have 8 in them.
We are facing the biggest challenge that this world ever seen right now, which is COVID. And Thermo Fisher is in the best place possible to fix it. Serving science is about making a difference in the world.
It goes back to the culture that we have at Thermo Fisher, being able to provide solutions and tools and expertise for our customers.
For the organization, it was being flexible and adaptable and being able to move with intensity.
I have never seen a more dedicated, more talented, more determined group of people to get this right.
I feel that we've been building up to this moment to help the world to see the benefits of science.
It's all about science. Science is what's going to save us.
It might not be front and center, but without our products and services, it really wouldn't be possible. I've always felt strongly that I wanted to work in a place that did good, and the good we are doing has exceeded all of my expectations.
I feel like I'm part of a group of 75,000 superheroes.
When I see and hear leadership using terms like society's depending on us, it really has an impact.
The impact that we're having is not something far into the future, but it's affecting our families, our neighbors, our communities now.
And knowing that the company I work for is adding value to the greater good of mankind, I mean, that's amazing to say I work for Thermo Fisher.
Serving science means
being able to see,
Making progress,
moving ahead with conviction.
Being a change agent,
balance,
Serving science.
Doing the right thing.
All right, so thank you everyone for and thanks to Thermo Fisher Scientific for this promotional video. So welcome once again to all the distinguished participants who have joined us today. And as you all know, today is webinar number 15. Monthly theme is biotherapeutics development, and today's chemistry, manufacturing, and control session. So I would like to start, as always, we start by extending our thanks to all our organizers. So all the presiding chair and chair-elect of different committees, big thank you to all of them. Because of them, we could, the entire agenda is in place with such a world-renowned speakers and interesting talks in place. Is all because of all the efforts put in by the presiding chair and chair-elect. And the presiding chair and chair-elect are supported by a very active organizing committee. So we would like to thank all our committee members also in regulated bioanalysis, in discovery bioanalysis, and the committee members in biotransformation, and committee members of chemistry, manufacturing, and control also. So a big thank you to all our very active and committed committee members for their very active participation throughout the year to be able to bring up such an interesting agenda on board for APA India 2021 webinar series. So big thank you to all of them. We also would like to extend our gratitude and thanks to our sponsors Shimadzu, Cyex, Zepsci, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, Natco Pharma, Vida Clinical Research, Bristol Myers Squibb, Zoetis, Nanotemper, In Vitro Admet Labs, Origin Discoveries Technology Limited, Vuxi Aptac, and Loris Infosystems. So a big thank you to all our sponsors, and many of them have been associated with APA India chapter since its inception back in 2009. And so a big thank you to all our sponsors for their continued support. This year, we have also collaborated with the Life Sciences Sector Skill Development Council, which comes under Ministry of Skill Development and Entrepreneurship. So thank you to our collaborators. Very quickly, just a few announcements. Poster session already we have been discussing for quite some time now. So 13 November, the final poster session will take place. Each poster presenter will get 8 minutes of slot. In that, 6 minutes is for their presentation and 2 minutes is for Q&A. In total, we are going to have 9 poster winners. So for first prize, there'll be 3 best poster winners. For second prize, there'll be 3 best poster winners. And for the third prize, there'll be 3 best poster winners. So basically, 9 poster winners would be there. There'll be cash reward, there'll be a trophy, there'll be a certificate, and a special prize for the one winner, which will be selected by the committee, the judges, especially from the academia. That particular winner will get an additional prize and a trophy from Dr. Singh, Dr. Singh's trophy, because the Professor Dr. Saranjit Singh, the trophy is going to be named after him. So that particular winner will have a free registration for the next year in-person meeting, and his local, his travel and his accommodation, all those things would be covered under this. So that is one special surprise gift which we are just announcing today, apart from the 9 poster winners. So that's about the poster session. Also, at the end of each and every webinar, we are doing a small Kindle draw. So if you want to participate, please hang on there till the end of the webinar. And after panel discussion, we will be doing this. So do participate in that. Finally, for the Q&A, for all the attendees, your audio and videos are muted. So you can always use Q&A tab to post your questions for any of the speakers you may have. During the webinar, if we are not able to address your queries, even in the panel discussion also, then feel free to write to us at bostondb21@gmail.com, and we'll reach out to the concerned speaker on your behalf and get your doubts clarified. To participate in the poll at the end of the webinar, if you want to participate in the, if you want to be eligible for the Kindle draw. And finally, all the webinar talks of all the 18 webinar talks will be uploaded on Boston International YouTube channel. So do subscribe that, and later on you can refer all this talk in case if you wish to revisit those. So with that, I think without further ado, let me invite the moderator, Dr. Vishal, who is with us from Bristol Myers Squibb. He is going to be the moderator and the chair for today's session. So over to you, Vishal. Thank you.
Right. Can you hear me well?
Yes, very well.
Awesome. Thank you. Thanks, Deepak, first of all, for the invite in this session. And I'm very excited to be moderating this session with the series of 3 talks that we have, all the exciting speakers and the topics that we have on the agenda today. So what we have are really, focusing on the biopharmaceuticals in the CMC section. We'll start off with the exciting talk from Dr. Satish Singh, talking about challenges on use of polysorbate surfactants, the control strategy around this in making sure we develop robust products which we can have for the life cycle of the asset. This will be followed by an exciting talk from Dr. Ming Du from Thermo Fisher Scientific, whose talk is focused on characterization methods for novel modalities, biologics, ADCs, and vaccines, etc. And especially on application of mass spectrometry-based technologies for detailed characterization of these novel modalities. Last but not the least, in the session, we have a talk from Dr. Anu Uppal, who will be specifically focusing on application of USP standards on the biologics assay method development, as well as characterization for a variety of different methodologies. So looking forward to this exciting panel and an exciting session today on a variety of topics that span the biopharmaceuticals. So with that, I think we can introduce our first speaker, Dr. Satish Singh. It is my utmost pleasure to introduce Satish. As usual, it is certainly a delight to have him. He has a great background. Obviously, many of us know that he's a world leader in biopharmaceuticals, in many aspects of biopharmaceuticals, and he's currently the vice president of at Moderna Therapeutics, and has had previous roles in Lonza Pharma and biotech, as well as in Pfizer. Again, as I said, Satish is highly recognized as an international leader in all aspects of biopharmaceutical drug product, biologics, vaccine development. He has experience in ophthalmics, parenterals, and biologics, as well as small molecules, and has been involved in successes of varieties of biologics and BLA filings, as well as INDs, and in filings in other countries as well. He certainly has a very strong technical background, complemented by deep understanding of regulatory landscape. He has published several peer-reviewed articles, and also been involved in the expert committee that authored the USP chapters 787, 1787, 771, as well as 788. He was elected a fellow of AAPS in 2018, and his research interests in particular has made significant impact, include the areas of freezing of protein solutions, computational approaches to mitigate developability issues, and in particular on protein aggregation and immunogenicity. He's heavily contributed to investigation and understanding of particulates in parenteral formulation and in ophthalmics. Satish obtained his BTech in chemical engineering from IIT Delhi, and his master's and PhD in chemical engineering from Kansas State University. He also is an adjunct professor at Uppsala University, as well as at the School of Pharmacy at the University of Michigan, Ann Arbor. I would especially like to thank Satish for, on a personal note also, he's been keeping us with his work he has done in Moderna, developing the vaccine, which we have benefited from, keeping us safe during this pandemic that we had. And hopefully he'll get us out of this virtual world and have a conference in person soon. So thank you, Satish, and really excited to hear from you today.
Thank you, Vishal. Yes, we all hope to be out of this pandemic. Stay safe until we are out of there. And hopefully you're all vaccinated or soon to be vaccinated. It has been an interesting year and a half, to say the least. Let me see.
Yes. Let's hear from you on the surfactants.
Yeah.
Friend or foe?
Okay. Is it working? Can you see the screen?
It is, yes.
Shall? Yeah.
Yes. Thank you.
And it's clutter-free, right? Okay.
It is.
Yeah. All right. Yeah. Thank you, Vishal, for a very nice and kind introduction, and thank you, Deepak. I mean, yeah, this is not the same as being in Goa or in Ahmedabad or in Hyderabad and so on, or Pune. But we'll get there hopefully next year. So I'm going to talk about surfactants in protein formulations, an old and favorite topic of mine. And for anybody who's worked with protein biotherapeutics, they know the need to have the surfactant and also the pain in the neck that these things can create for us. So I'll jump into it. Oops. Yeah. I'm going to skip over the abstract. So to begin with, I'm going to introduce it like when you have a trailer for a movie as to why the heck are we talking about surfactants here. This is a somewhat old story. Ocrevus filed by Genentech. Actually, if you look in here and you think for a company like Genentech, actually CDER did not recommend approval of this product for CMC reasons. Well, actually they did approve it because of unmet medical needs, but they did not. The CMC reviewer was not very happy with this. And part of the reason, if you look through their summary of the review, you find concerns regarding stability of the drug product. They wrote, right, the degradation of polysorbate 20 was seen in historical and process validation batches. They saw visible free fatty acid particles on process validation batches on stability at late time points. And an interesting question, shipping validation did not include evaluation of aged drug product. That is to say, drug product that had degraded polysorbate 20 in it. So they got actually a fairly short dated expiry period of 15 months. So why did this all happen? And this is all, this is part of the topic, some of the topics that I will cover in my talk here on surfactants. And in the interest of time, I'll skip over this outline of the talk, basically introducing the surfactants, especially the polysorbates as the most common one, and their properties, function, degradation, a couple of slides on safety and control strategy. Okay, so why do we, if you're a formulation scientist working in the protein world, you know why we add surfactants. Surfactants are added to protect against interface-induced aggregation and particle formation. So interfacial stresses are frequently encountered during many stages of production, shipment, and use of the product. By this, I mean air-liquid interfaces, such as during shaking or during the shipping, and you can connect to the Ocrevus review. You saw a couple of slides back. Ice-liquid interfaces, surface interactions with manufacturing equipment, with use, with clinical materials, etc. So anywhere where there is an interface, there can be an interaction of the protein with that interface, causing denaturation and aggregation, and surfactants protect against this. So interfacial stresses lead to protein instability, such as adsorption on the surface, aggregation, or precipitation. And surfactants, such as the non-ionics, polysorbate 820 and 80, effectively protect proteins against these interface-induced stresses and adsorption. That's all we need to say about this. That's the main reason why we add these. And for almost in all 100% of the cases, these are almost always non-ionic surfactants. And I would venture they're almost always polysorbate 20 or 80. Although there are now a couple of products with poloxamer in there. So just staying with the most common one, the polysorbate, what is the common structure of these polysorbates? There are two versions of the polysorbate that is most commonly used, polysorbate 20 and polysorbate 80, tween 20, tween 80 as also. And they're basically they are sorbitan esters, polyoxyethylene sorbitan esters. Here's the hydrophobic part of them. In this polysorbate 20, it's supposed to be the lauric acid based fatty acid, and in polysorbate 80 is the monoureate or oleic acid based polysorbate. And you will have to note that the oleic acid has a unsaturation here, a double bond in it. So here's the hydrophilic end, the sorbitan ester, the polyoxyethylene sorbitan, and then the hydrophobic end, the fatty acid. That's the general structure of these. However, they are not uniform. So polysorbate 20, although primarily composed of lauric acid, will have components of other acids in them. So ranging from C6s to C18s, and then smaller amounts of the the longer chain fatty acids. And similarly for polysorbate 80, tends to be more of the longer chain fatty acid, with oleic acid being the main component. So unlike this diagram with a very fixed structure, you can see that there can be a mixture of compounds that make up the polysorbate 80. And they are listed in the pharmacopoeia, USP, EP, JP, and also the Chinese pharmacopoeia. And the one thing I want to point out, although I'm sorry, this is very small, the USP, EP, and JP are harmonized in terms of the amount of oleic acid in them, but the Chinese pharmacopoeia tries to go towards a more monooleic form of the polysorbate. So it has a minimum of 98% oleic acid requirement in the polysorbate 80. Doesn't mean it's more pure. People sometimes say, "Oh, this is more pure." No, this is not more pure. It's just it has a higher percentage of oleic acid. Purity is not determined by the amount of fatty acid in it. It's just the composition that's differently defined in the Chinese pharmacopoeia. So here's an example of the fatty acid esters in polysorbate 20, taken from multiple batches and from one supplier, actually from two suppliers. And you can see polysorbate 20, a lot of lauric, myristic, palmitic, stearic, oleic acid. So there is a distribution of fatty acid esters, but generally consistent between batches. And similarly for polysorbate 80, again, a distribution of fatty acid esters, but consistent between batches, which is what we want for the most part. So if you look at it, what else may be present in polysorbate 80? So polysorbate 80, from the process of synthesizing it, can have polyoxyethylene sorbitan, so sorbitan with oxyethylene esters on it, mono di triesters, isosorbide, so a dehydrated form of sorbitan. So there can be a whole host of species, including free fatty acids, all of these arising from the process of making the surfactant. And then there can be other products arising from hydrolysis or oxidation of this material, such as free fatty acids, peroxides, aldehydes, ketones, fatty acid esters, etc. And an important thing to note is that these polyoxyethylene sorbitan, the one without the fatty acid, the non-esterified version of this, has no surface activity, so has no protective effect, because the protective effect arises from its surfactant behavior. So to summarize, polysorbate as an excipient is highly heterogeneous. Monographs allow a wide range of fatty acids in its composition. However, what's most important for us as a user is that there is consistency between batches of polysorbate. And it's important to have tools to analytically characterize the material we receive. And this is in itself a complex topic, and I am not an analytical specialist. But just because of the number of species present in these excipients, they require a multitude of methods to characterize them. And they also vary between batches. So this is another important factor. So what happens is, if you end up using a surfactant from one manufacturer, you tend to stick with them because if you change, it becomes a critical excipient that has to be changed, and it requires all the considerations, perhaps extra stability studies, and maybe more safety studies if that's the case. So coming to the function of this material, we already covered this earlier. The function is primarily to stabilize the protein against interface-induced damage, against freeze-thaw, agitation, shaking. People talk about it also stabilizing against lyophilization. I have not seen so much of that. There can also be prevention of loss of the active by surface adsorption. So if you have a protein with a low concentration product, or a low concentration product, which you could lose to the container walls, this can prevent that adsorptive losses. And I've also seen it stated as that it's an reconstitution aid for lyophilized product, i.e. it can make reconstitution easier and faster. We have not seen this readily, but there may be instances where this can also be a benefit. So how do proteins, surfactants stabilize the protein? We already talked about it. The mechanism is for each a specific protein is not exactly known, but it tends to be the competitive interaction of the surface active component to the interface. So that means instead of the protein going to the interface, as they would tend to do, because most proteins have a hydrophilic, a polar behavior, the surfactant goes to the interface, prevents the protein from occupying the interface, and therefore eliminates its presence in this interface and reduces the risk for denaturation. So so this cartoon basically exemplifies the behavior that is typically assumed for the protective effect of the surfactant. There can be another effect wherein the surfactant adsorbs onto the protein and makes it more hydrophilic, and therefore prevents it from going to the interface. It also prevents close approach between proteins, but so this would depend upon the type of protein, but one can also imagine that this kind of adsorption has greater risk, because if there's too much binding, the surfactant can actually cause the protein to unfold. So depending on the type of protein, it can be in a surface interaction protection or this binding protection, depending on the polarity of the protein itself. But surfactants can have a dual effect or an inverse effect. In this study, they looked at the PEG-GCSF and impact of polysorbate concentration on the aggregation behavior, as they had here's increasing polysorbate levels, and when they kept the solution just quiescent, that means without agitation, for 59 weeks at 30 degrees centigrade, they had increasing aggregation with increasing polysorbate 8, polysorbate 20. But when they used agitation, there was protection afforded by the polysorbate 20. So you can see here there's protection, just as you would expect from by the polysorbate against aggregation due to agitation, but it can also cause aggregation in itself. And this may be, this could be due to oxidation induced by the oxidation of polysorbate and I'll cover that in a couple of slides later. Or it may be this reduced free energy of folding through interaction with the hydrophobic sites of the protein itself, as I showed in the previous slide. If the polysorbate interacts with the protein, it can cause, or too excessive interaction of the protein can cause unfolding and aggregation. So polysorbate doesn't come without its risks, is what this slide shows. So now coming to a critical aspect of understanding polysorbate is its degradation and the impact on protein quality attributes. So polysorbates, by their very nature, are labile in the sense that they, as raw materials and in product, they can degrade due to multiple factors such as light, especially UV light, temperature, oxygen. If there are metal ions in the solution, there may be an impact of the pH, and if there are certain types of leachables potentially, and then also something called lipases or enzymes that can degrade or interact with these surfactants. So as you can see, a multitude of factors that can impact the stability. So, but there are two main mechanisms: the hydrolytic degradation mechanism, which primarily attacks this at this ester bond and creates free fatty acid and polyoxyethylene sorbitan. So that means polyoxyethylene sorbitan here, and then the free fatty acid from here. And And with this kind of a degradation, the surface active species is lost because you've lost the hydrophilic and the hydrophobic parts have separated. So you don't have any surface activity left. On the oxidative mechanism is a lot more complex. It's a free radical based mechanism. It may have a lag phase. It can attack at many of these oxyethylene sites in multiple places. And if the fatty acid itself has an unsaturation, and therefore it yields a whole host of species, fatty acid esters, free fatty acids, and these can then in turn degrade to form aldehydes, peroxides, acids, alkanes. So a multitude of species. But the important thing is that a number of these species retain their surface active behavior. So you can have oxidative degradation, but you can still have protection from the surfactant. And the oxidative behavior, just as an example, oxidized polysorbate 80 has the presence of peroxides, and these peroxides can then interact with the protein to cause protein degradation. And this is what this slide is showing. Improperly handled polysorbate 20, in this case, resulted in an oxidation of the tryptophan through the peroxides. And this one talks about particle formation due to free fatty acids from the polysorbates. If you remember, the hydrolysis of the polysorbate leads to the formation of free fatty acids. Free fatty acids, if there are enough of them, will exceed their solubility in the buffer or in the solution, and will come out of solution and be looking like a snow globe of particles. And if you remember back to the Ocrevus review, they talked about particles forming in the product over stability, and this is the kind of particles that would be seen over time, just because of the free fatty acid. So surfactants can be a source of particles in biotherapeutics. So what are the causes? And some of the more recent, perhaps in the last eight or so years, this has become much more recognized that host cell protein enzymes can mediate the hydrolytic pathway that generates free fatty acids. So this is a concentration dependent effect. The higher the concentration, the more likelihood of a higher concentration of host cell protein enzymes coming through purification and then having an impact on the formation of the free fatty acids. Here's an example of one such study where they took a MAB pool, a UFDF pool, and monitored for the presence of lauric acid and myristic acid as signals for degradation of the polysorbate. And when they added a compound called orlistat, which is an enzyme inhibitor, they were able to suppress the formation of lauric acid and myristic acid. Without that, these free fatty acids increased with incubation time, indicating that in this pool there was some lipases present. And later on in the paper, they described the presence of lipoprotein lipase, lysosomal acid lipase, and palmitoyl protein thioesterase 1. So a multitude of lipases arising from the host cell protein itself. Okay, and one last thing, polysorbates can be lost during processing. So if you're using filters and tubing and so on, it's one thing to be concerned about because surfactants adsorb to surfaces such as filter membranes and tubings. So if you're going to fill a product, you will have to ensure that the filter and the lines get saturated before you start the filling process. Okay. Safety of polysorbate. So surfactants have some tolerability issues. Now, if you look at the literature, and if you look at the literature on surfactants, high levels of surfactants are used in chemotherapies with small molecules. A lot of these molecules are hard to dissolve, have low solubility, and therefore a lot of surfactants are used to dissolve, and therefore extremely high levels of surfactants are dosed in many cases. So a lot of these side effects, tolerability, and administration issues that you see in the literature actually relate to extreme doses of surfactants being given in chemotherapy, for example. On the other hand, with biotherapeutics, when we use these, we use them at 0.1, 0.2 mg per mil levels, so the doses are never that high. Nevertheless, it's important to know the surfactants, their safety signals. So systemic administration of polysorbate 80 can lead to transient hypotension at fairly high levels. Here it is 300 milligrams in 10 minutes. And in the case of dogs, greater than 5 mg per kg dose to dogs leads to tachycardia and so heart effects. This reaction is associated with histamine release, so it's an immune or allergenic reaction. Similarly, there can be anaphylactoid systemic reactions such as urticaria, hemodynamic compromise, chest tightness, respiratory symptoms. This was actually reported for omalizumab patients and in cases with IV multivitamin, but here again the doses were very high. There can be acute hypersensitivity reaction through complement activation. It's potentially attributed to the degradation products, for example, oleic acid in polysorbate 80. It's been shown that degradation products of polysorbate 80 can upregulate the expression of CD86 cytokines by dendritic cells and therefore lead to these hypersensitivity reactions. Again, they are not present in everybody. It is a susceptible situation of susceptibility. Contact dermatitis may be caused by oxidation products such as aldehydes. Another sort of related issue can arise because surfactants are solubilizers, and it has been reported in the literature, and in the case of one product, it's also mentioned in the package insert that this polysorbate 80 can extract the phthalates from PVC-based infusion sets. So these phthalates are used as plasticizers in plastics in infusion sets, and polysorbate 80 can extract them depending upon how much, the extent of the contact time, etc. So here this is cumulative amount of diethyl hydroxyphthalate released over a period of 5 hours, depending upon the amount of polysorbate 80 and the flow rate. And we know that phthalates can function as oxidants for the protein. So if an infusion product is made in these bags with polysorbate 80 and kept for a while before being infused, you could have a risk for oxidation of the protein due to the extraction of DHP. Control strategy. So polysorbate is a critical excipient in the drug substance because we use them in biotherapeutics. We use them in fairly low levels, but we know they are important to have there due to this important stabilization function that they perform. So it is a critical excipient, and therefore the FDA may require testing and quantitative acceptance criteria for levels of polysorbate 80 as part of the in-process controls or in drug product release specifications. This is an actual feedback from the FDA that I've quoted here. So you have to show a proper control strategy for the polysorbate 80. You have to know how much you add, you have to know where it goes, you have to know its degradation pathway, and so on and so forth. And if you have to put it on release specifications, that's an extreme. And then if you have to monitor it on stability, like again in the Ocrevus case, that makes for a difficult analytical task. But a good control strategy comprises of all stages of at the raw material testing and qualification, product characterization throughout development, defining what are the process parameters and where will you control this in the process of manufacture, for example. Will you have it? Will this be present on release? Will there be specifications on the polysorbate content? And of course, on stability testing. So, depending upon the individual product, its susceptibility, a variety of control strategies may be required. Obviously, one would want to avoid putting it on specifications and avoid having to test on stability, but in certain cases, it may not be avoidable. But one of the most important and the simplest things to do is to have a proper raw material control, a proper protected storage and use of polysorbate, so that the raw material is of good quality when it goes into the product. A degraded polysorbate is not good for the product, and it's bound to cause trouble later on. So a bit of prevention by using, a new container or not reusing old polysorbate 80, good nitrogen overlay, good handling practices will go a long ways to prevent issues with polysorbate 80. So with that, I would like to summarize. Proteins generally love to have non-ionic surfactants. Without them, they will aggregate, destabilize. But proteins can also hate surfactants because they will cause oxidation and so on. As formulation scientists, we love these, but at the same time, we hate the issues that these things can create. Polysorbates are a complex mixture. They degrade. Degradants can impact protein stability, can lead to particulates and peroxides, oxidation. They can be lost during processing. So that's why they are both friends and foes of ours as formulation scientists. To summarize, surfactants are complex and generally poorly defined excipients. There are new analytical tools coming out now that are improving this to an extent. They play an important role in the product, but their activity is complex. They can be detrimental to stability. Raw material quality control is critical to their protective activity. Good analytics with a good analytical toolbox and expertise is critical to develop. And an overall control strategy should be established based upon the criticality assessment of its function. With that, I would thank you for your attention, and I hope, Vishal, I've been on time, but hopefully. Can we take questions now, or we can wait till later? Thank you.
Yeah, no, Satish, that was an excellent talk. There are a few questions that we can take now, and then we can take a few during the panel discussion. So the first one is, is both PS20 and PS80 are widely used in protein formulation? Are they interchangeable? Are there any specific principles when we select which one to use for a protein formulation?
Well, in principle, they are interchangeable. I've not seen anything published or experienced where you say, oh, polysorbate 20 is better than polysorbate 80, or vice versa. Polysorbate 20 has a slightly higher CMC micellization concentration, but I've not really seen any significant benefits. Polysorbate 80 has this unsaturation in the oleic acid, which may be considered as having maybe perhaps a bit more risk for oxidative degradation, but that's not necessarily the only spot that it can oxidize. So on the whole, what I've found is that what happens is most companies get into this thing. Well, we either use polysorbate 20 or we either use polysorbate 80. That once you start using it, you have procurement of the raw material and so on. Everything is in place. You just end up using it. I've not seen any. For example, Genentech, Roche always use polysorbate 20. I don't think there is any great scientific reason other than that that's what they started with. So they are interchangeable in the sense that, yeah, I suppose you could use a different one, but in the end, we end up using the same because of the fact that it's a critical excipient. You must have controls, analytics. So it's just simpler to keep the same excipient. Was there another part to the question, Vishal?
Yeah. No, I think you covered
got
most
it.
of it.
Yeah. Thank you.
Yeah,
Vishal, Ravi here. Am I audible? I have a couple of
yes,
questions.
you are. Yes.
Yeah. Thank you, Dr. Excellent presentation, Dr. Satish. So in-depth, deep dive in the polysorbate, which is really a important component. So one of the question is nowadays we, in the sense there are polysorbates which are highly pure. That means the oleic acid content is extremely highly pure when compared to a general polysorbate. So what is your experience that using highly pure component would be better to control all these aspects, or the behavioral aspects of these polysorbates?
Yeah, so I do want to maybe when you say highly pure, I do want to make sure that we understand when we say purity, just because it is 98% oleic acid doesn't make it highly pure.
Okay.
Highly pure means highly pure means it doesn't have peroxides in it, doesn't have free fatty acids in it. That's what I would consider highly pure. When you have, say, 98% oleic acid as opposed to 58% oleic acid and 20%, that doesn't make it pure. I mean, it still meets the pharmacopoeial definition. It's just more mono-oleic or so on. Yeah, so I always try to correct that, because purity is against impurities, and having, say, you saw in the USP and the EP definitions, a certain amount of lauric acid is allowed and a certain amount of stearic acid is allowed and so on. So that doesn't make it impure. So coming back to the other question about whether there is a functional difference between oleic acid, high oleic, I prefer to call it
formulational behavior perspective, like I mean, aggregational formation and other things. Yeah.
Right. So if will a high oleic acid polysorbate 80 be better than the standard polysorbate 80, for example, right?
Okay.
So the Chinese pharmacopoeia polysorbate 80 versus the USP polysorbate 80. Yeah. There is a paper that was recently published by the folks from Coriolis that shows that both are equally effective, that there was no functional difference. I had a slide on too, but I didn't put it on for time reasons. But in that paper, they show that at least in their functions, in the typical functions, there was no difference in the behavior. To some extent, in my own belief, I obviously haven't got this study very well, and it may take more data or more careful studies. I feel that a wider distribution of fatty acids may give a benefit in the sense that you can cover a variety of, you have a variety of species that migrate at different speeds, have migration at different rates to the interfaces, but I don't have any specific data for it. But the only data that I've seen published says that there is no difference between those two grades.
Thanks, Doctor. So clarification. My last question is related to
Ravi.
Okay. Yeah, thanks.
I'll be here. I'll try to stay awake and.
Absolutely. Yeah. I understand the time constraint. Yeah. Sorry.
All right. Thank you. Thank you, guys. Yep.
Yeah. Thanks, Satish, again for an excellent talk.
Thank you, Vishal. Yeah.
All right. So, so with that, we would like to facilitate Dr. Singh for his excellent presentation, the talk on the surfactants and protein formulation, friend or foe, from APA India. So, thank you.
Thank you, Vishal. Thank you, Deepak. Thank you, guys.
Thank you, Satish, and just wanted to reconfirm that after third talk, we'll have the panel discussion. So if you're around, there are quite a few many questions for you, so we can take up those questions during that time. Thank you.
All right. Yeah, I'll be around.
Over to you, Vishal.
Sounds good. So we are on to the next presentation, and I would like to welcome again Dr. Min Du from Thermo Fisher Scientific. Thanks for staying up late with us. And so Min comes from, like I said, Thermo Fisher Scientific. She is a senior manager for the pharma and biopharma applications at Thermo Fisher's scientific chromatography and mass spectrometry division. Min and her team are responsible for utilizing state-of-the-art liquid chromatography and liquid chromatography mass spectrometry technologies to develop methods, applications, and solutions to meet pharmaceutical and biopharmaceutical developers and manufacturers' need to develop robust products. And especially, I'm really curious to hear from her around her talks on the new modality aspects. So to you, Min, welcome again, and thank you for looking forward to your talk.
Thank you, Vishal. And I'll start to share the screen. Please let me know when you start to sit.
Yeah, we see it, Min, if you can put it in. Yeah, perfect. That's great.
Okay. Okay, thanks for the kind introduction. And I would like to first thank the organizer to provide this opportunity, and everyone taking time on a Saturday morning to attend this event. Today, I would like to share some of the recent work we have done to apply the state-of-art high-resolution LC-MS technologies to characterize new biologic modalities. It's not a surprise, but most of you probably already know in recent years, we not only saw the increasing approval and the continuous strong growth of monoclonal antibodies in biologics drug market, but we also witnessed the development and commercialization of many novel therapeutic modalities such as ADC, multi-specific antibodies, nucleic acid-based drugs, including oligonucleotides and their hybrid conjugates, and most recently for mRNA-based vaccine therapeutics candidates, and as well as gene therapy. And many of these next-generation biotherapeutics are more complex and aim for fulfilling unmet medical needs with accelerated regulatory approval designations, thus present new challenges for the development, manufacturing, and commercialization. All those challenges requires more advanced analytical solutions to fully characterize and monitor their critical attributes during their development and manufacturing with increased productivity to ensure quality, safety, efficacy of those drugs in development. So today, we'll, with the time restraint, we'll only focus my topic on characterization of mRNA molecules and the viral vectors for gene therapy. So the first analytical solution I would like to discuss today is our recent developed end-to-end workflow for mRNA sequencing by high-resolution LC-MS technology. So messenger RNA mRNA has recently become a new prospective class of vaccine and therapeutic products. The potential of mRNA technology for rapid vaccine development has recently been highlighted and well demonstrated by the successful development of COVID-19 vaccines from Moderna as well as Pfizer-BioNTech. Current analytical methods to characterize those molecules are limited, and the development of new analytical method for the analysis of large mRNA drug products is in much needs. Here I'm showing a very simplified schematic to demonstrate the in vitro transcription process to generate mRNA from plasmid DNA. This can be done in very small quantity for research purpose, as well as later on can be scaled up for cGMP production. The assay I'm going to focus on today is actually mRNA sequencing, while separate LC-MS-based assays can be used to determine 5' cap efficiency as well as 3' poly-A variants. but today I'm not going to cover those two assays. Compared to the conventional sequencing technologies that requires amplification and convert RNA back to cDNA libraries that do not direct sequence the mRNA molecules, a LC-MS-based method can provide faster and direct sequence information, as well as the capability to assess modifications. However, unlike analyzing smaller oligonucleotides such as antisense oligoacyl and double-stranded siRNA, large mRNA are not suitable for direct intact mass analysis. Therefore, digestion of the molecules with appropriate indoribonucleotase RNase enzyme prior to LC-MS method for oligo identification is required. You can see there are some challenges that need to be addressed. There are only 4 constituted bases for the mRNA, and thus ideally we wanted to generate after digest longer oligonucleotides between 10 to 60 nucleotides lengths that are more ideal for good MS fragmentation, but also with increased sequence specificity. Because if you cleave them in very small lengths, you will have those sequences coming from many different places of the mRNA sequence. Thus, you cannot have the confidence that those other sequences can cover all the areas. So for example, showing here, there's no recognized specific nucleuses which will provide the correct fragmentation length. For example, a very common nucleus T1, it cleaves at the HG residue for the single-stranded mRNA. Complete cleavage will give fragments that are too short, so thus require a partial digest. But the enzyme is very potent, so it works very fast and it's very difficult to control and stop effectively. Thus make the partial digest optimization and control very difficult. Also, in solution digest usually can cause a contaminate of the analytical column within the nucleus for the subsequent LC-MS analysis. Last, but also very important, there's a lacking of optimized LC-MS methods, as well as automated software solutions to really help the scientists to develop and optimize this method, especially for the software solutions, because as you can see with the larger mRNA molecules, the digested oligonucleotide can be presented with very complex mixtures. Manual interpretation is a mission impossible to sequence this molecule. Here we demonstrated the newly development of a specific end-to-end RNase mapping workflow optimized for the analysis of our mRNA. This workflow starts with automated sample prep using magnetic beads with immobilized RNase. Over here, we show the RNase T1. We also achieve great separations of the complex oligonucleotide mixture using the DNA pack RP column, specifically designed for oligo separation. And on a totally biocompatible Vanquish HPLC system, analyzed with Orbitrap Explorer's mass spectrometer in conjunction with automated software analysis using Biopharma Finder that match fragment MS sequence data to the mRNA sequence, resulting in high sequence coverage for this total workflow. First, let's take a look at the endonuclease digest. As I mentioned before, we need a partial digest to get fragments that are big enough to be unique. Compared to the in-solution digest, RNase immobilized on magnetic bead surface offers the opportunity to easily control and optimize this digestion process and will not cause contamination for the LC column in the subsequent analysis. As shown in this slide, you can start with your mRNA sample in solution, simply introduce the bees to start the digest at the set temperature. And by removing the bees from the solution, you end the digestion immediately without the need to stop the RNA's activity in solution. We currently offer two commercialized kits, RNase T1 and RNase A magnetic bulk kit, and as shown the part number in this slide. This approach can be easily automated using the Kingfisher dual prime system if you opt in for the automation. But if you don't want to be automating, you can usually just do, shaker, heater, shaker, and you can simply do this process as well. We do recommend the automated approach because it actually can aid in reproducibility and the control of the time. So here the next step is LC-MS analysis. Here you can see the chromatogram of the mRNA of a spike protein, the COVID-19 spike protein mRNA digest using IPRP LC-MS method. The DNA pack RP columns are specifically designed and well accepted by our industry partners for oligonucleotide separations. And it provides superior separation for both the small and the large oligonucleotides. In this highlighted retention time window from 3.5 minutes to 33 minutes, you can observe the high resolution separation of the oligos ranging from 11 mers to the 48 mers. The Orbitrap Explorer's MS system are also well suited to analyze the complex mRNA digest samples with many negatively charged ions associated with each oligonucleotide species from smaller size to the larger size, providing the high scan speed and exceptional sensitivity and unrivaled high resolution accurate mass full MS and MS data is essential to get good data. So next, let's take a look at the mRNA mapping analysis in Biopharma Finder software. The identification of the oligonucleotides analysis launched last year has been expanded to include the in silico mRNA digest. As you can see that you can select from common RNAs on this list, and you can also add customer RNAs. And the software will identify the mRNA pieces using MS data or full scan MS1 data and provide a sequence coverage map. I would like to highlight the main benefit of the component detection in this software. These mRNA samples are very large and very complex when digested. Because the software was extremely sensitive and reliable, with reliable component detection, the digested mRNA fragment can be detected in a very complex mixture at a very low level. My cursor somehow went wrong, sorry. Improvement has been also added for manual validation of MS2 spectra in the oligonucleotides analysis workflow. The user can view the theoretical fragment as you can see on top of the right-hand panel and compare that to the real MS2 spectrum with annotation and the sequence coverage map shown in the middle. This will allow for deeper investigation into the matched identification. In this example, we can see the baseline separation of two isomers with identical base composition that can be easily identified by MS data with base-by-base sequence confirmation, demonstrated the need for high-quality MS data for unambiguously identify those mixture oligonucleotides. The component detection not only look for all the individual masses in the full MS to identify potential chromatographic peaks, but it also performs intact mass deconvolution, which allow you to identify and associate all the charge states present for each component or digest fragment present in the chromatogram. These identified components are then matched to their corresponding MS scan for further identification confirmation. Here we show the full sequence coverage achieved for a rather larger oligonucleotides of 48 mer. Combining the high-quality MS and MS data generated from the Explorer's MS system, in this case, you can see that the full sequence coverage was achieved for this large oligomers with multiple charge states sequence confirmation to really boost the confidence of the identification. Next, we want to show the power of partial digest for increased sequence coverage. Compared to the complete T1 digest, as you can see on the right top panel over here, the green highlight is a sequence for identified and the red one are not identified. So if you simply compare those two, you can find that the partial digest have majority of identified sequence coverage versus opposites in the complete digest. So for the complete digest, we got about 14% sequence coverage. And for the partial digest, we got about 90%. With the partial digest, we don't observe any non-specific cleavage, and the overlapping fragment are very useful for the provide unique and high quality fragmentation information. Versus if you look at the result from RNase-T1, because many of them are smaller species, only one of the longer mers, 21 mers, are useful fragment for sequencing mapping with a full digest for this particular session. To summarize, the mRNA sequencing workflow works well. From our experience, several mRNAs now tested with good results. Partial digest can be optimized, and the oligo fragments can be well separated, and we can achieve a high quality MS and MS data from the Explorer's Orbitrap system. And with Biopharma Finder software, it really can aid the productivity for optimization of this sequencing data analysis and method development. With that, we conclude the mRNA session. Next, we would like to switch gear and look into AAV capsid protein characterization. According to at least one of the market report, gene therapy market was estimated to reach US $21 billion by 2027 globally, and with a CAGR of 30% year to year from 2021 to 2027. AAV is a very versatile viral vector technology that can be engineered for very specific functionality in gene therapy applications. It can be used in a wide range of clinical applications in multiple diseases due to its unique biologic and biophysical properties. To date, AAVs has been shown to be both safe and effective in preclinical and clinical settings. There are two approved gene therapies on market, Luxturna and Zolgensma from Spark and Novartis, respectively. AAV capsid is comprised with three structural viral proteins, VP1, 2, and 3, assembling the viral particle at a ratio of about 1:1 to 10. All three VP proteins share the same C terminus with N-terminus extension for VP2 and VP1, respectively. The capsid proteins play important role in cell binding, internalization, and trafficking, and are required to be fully characterized to ensure the safety, quality, and efficacy of AAV drug products. In addition to conventional assays such as qPCR, AUC, ELISA, SDS-PAGE, LC-HRAM-MS has emerged as a fast and direct analytical method for AAV particle characterization. In the following slides, I will demonstrate the information-rich data you can obtain from 4 workflows shown here, including the denatured intact mass workflow, as well as peptide mapping, HCP, and the top-down workflow. All those work has been performed on a single LC-MS system with Vanquish Horizon UHPLC system coupled with Orbitrap Explorers for 80 mass spectrometer. And all data analysis were also carried out using Biopharma Finder software. 1 millimeter columns were used for improved sensitivity without sacrificing the assay robustness. First, you can see the high quality intact mass data can be obtained for 3 viral proteins, and also the truncated form present at a very low level of VP3 proteins. Major modifications such as acetylation and phosphorylation can also be identified and relatively quantified at the intact level. The VP protein ratio was determined by the fluorescence detection, and the fluorescence chromatogram was shown in the center of this slide. And you can see the ratio of the VP1 to 3 proteins are determined as 1.3 to 1 to 10, very close to theoretical ratio. High quality full MS raw spectra were obtained for all 3 proteins, and the deconvoluted mass accuracy are all well below 3 ppm, even including the low abundant VP1 and the VP2 proteins. One of the major challenges of AAV capsid protein characterization is the limited sample amount for many different analyses needed to be done to fully characterize the capsid. Here we demonstrate even with a very low, much lower sample loading on column down to 28 nanogram, you can still get excellent quality spectrum with accurate mass measurement for the low abundant VP1 proteins, really provide a very sensitive and confident assessment at the intact level. For those low abundant VP protein truncated forms, in addition to the accurate mass measurement with full MS data, HCD top-down analysis can also be used to further confirm the sequence assignment that give you additional evidence of the sequence confirmation and the identity of the species. Next, let's move to the peptide mapping analysis. We compared the trypsin digest, which is typically used for protein peptide mapping analysis, versus the pepsin digest. For AAV peptide mapping, we find that the pepsin digest will provide the complete sequence coverage of 100%, versus trypsin usually get about 80% sequence coverage. Thus, we choose the pepsin digest for the sequence confirmation as well as PTM analysis. All 3 VP proteins and terminus can be confirmed with high quality MS data, also shown over here. And their C terminus are the same sequence. Post-translational modifications in AAV viral capsid are known to potentially affect their function, such as reported deamidation of amino acid on the surface of AAV capsid can lead to charge heterogeneity and impact the transduction activity. With peptide mapping analysis, a comprehensive PTM identification and relative quantification can be achieved even for very low abundance species. As we've shown here, across all 3 modifications, such as the serine phosphorylation, methionine oxidation, and asparagine deamidation, they all well below 1%. And you can see a very high quality MS spectra for each peptide, really aid in the confident identification of those modifications. The bottom-up analysis can also be used for HCP wholesale protein identification and relative quantification. We also compared the two digestion protocols, and for HCP analysis, actually trypsin digest works better than the pepsin digest. Thus was used for HCP analysis. And we performed this analysis for both crude AAV harvest and compared that to the post-purification sample. In the crude harvest AAV samples, 817 wholesale proteins were identified versus 30 identified in the purified samples. We filter our result at least that has 3 unique peptide identification, then we confirm the identification of the HCP protein. With the high separation efficiency and the greater sensitivity offered by this micro UHPLC MS-MS approach, the MS-MS data were obtained over a wide dynamic range. As shown in the top left side figure, you can see although the peaks of detected peptide from this HCP, which is labeled in purple color, they all buried in the background because of their low intensity signals. However, even with those low abundance signals, the Orbitrap Explorers 480 mass spectrometer still triggered MS-MS accreditation and generated high quality data for confident peptide identification. With a spiking standard, we can estimate the level of this HCP is about 3.6 picomole per mil. So it's a very low level in the crude harvest AAV6 samples. In addition, the integrated peak areas values for those for this low abundant peptide demonstrated good reproducibility. For example, over here you can see among 3 triplicate injection, the percentage of CV is about 11%. So very reproducible approach. The percentage using Biopharma Finder software, it can also help you easily track the clearance of each identified HCPs. As you can see shown in the right-hand side figure, there are 6 HCP proteins trending plot with 3 replicase injections for each sample shown over here. Very easy for the users to track the clearance rate. With that, I would like to summarize. Using this approach, we can enable complete application solution for AAV capsid protein characterization through all 4 workflows: intact mass, top-down peptide mapping approach, including HCP analysis. With the excellent sensitivity, we have excellent sensitivity to detect low abundant intact viral proteins with great mass accuracy, allowing rapid AAV capsid protein characterization, even with limited volume, low titer AAV samples. The confident protein sequence confirmation of the truncated proteoforms can be further confirmed using the top-down approach. By using pepsin enzymes, we can achieve 100% viral protein sequence coverage with a single LC-MS run. The high sensitivity and the high quality MS and MS-MS data offered by the developed method also allow the high confident identification and robust relative quantification of low abundant site-specific VTMs and wholesale proteins. And with all the workflow presented today here, Biopharma Finder 4.1 software provide a single software solution to fulfill the need for complete capsid protein characterization approaches. Here are some additional resources you can explore more detailed information, including 2 application notes on AAV viral protein characterization and a biopharma webinar series featuring multiple webinars from our customers as well as internal speakers for new modality analysis. Last but not least, I would like to acknowledge and thank all my colleagues contributed to the work demonstrated here, and also our collaborator, Professor Mark Dickman at the University of Sheffield. And thank you all for your time, and I would like to take some of your questions. Thank you.
Okay. This is excellent. Excellent work. That's very impressive to see, what kind of resolution and sensitivity you get with these methods. So one question I have to you is, have you done, similar work with lentiviral vector, for instance? I'm talking in the context of the AAV. I mean, my question really is coming from the fact that is there a big challenge using enveloped viruses with some of these techniques or not?
So most of the work so far we've done are focusing on the AAV viral vector characterization. There are increased need to characterize actually lentiviral vectors, and that's actually in our work plan. And we started to work on the HCP analysis, but not on the viral vector characterization. But also that's a question to the industry partners is for AAV characterization, I think there's better understanding what kind of areas people want us to focus on. For lentiviral vectors, most of the cases, they are like intermediate, right? So they are used more in the cell therapy. And what are the requirements when you are an intermediate product, not the final product? So that's actually a question turning the directions to our customers and really understand what is the requirement and what we can help our partners to succeed in that field. But that's definitely immediate project in our project plan.
Yeah, no, I think what you mentioned about deamidation potentially impacting the transduction efficiency and those things, I think that could be pretty interesting from a lentiviral vector perspective also, because that variability is something one might want to avoid in transduction efficiency if you have those kind of variations and things.
Yeah, I think there's definitely some common ground between the viral vectors. And the lenti system is also very popular system because it can really encompass larger, DNA molecules compared to the AAV. The AAV still have its limitation. Every system has its own limitation. Either a non-viral vector versus viral vector. But it's a very exciting to me. Is throughout my career so far, I never saw so much like excitement and the diversity of development of the different modalities as right now. So it's, as analytical scientists, it's very, very exciting to actually face those challenges and try to help to solve those challenges as well.
Sure, absolutely. Thank you so much. Yeah. I think in the interest of time, we can keep the rest of the questions for the panel discussion. So once again, would like to facilitate our speaker, Min Du. So Dr. Min Du, thank you again for an excellent presentation and wonderful work.
Thank you.
Thank you, Min. And then you is also going to be there for the panel discussion. So we really appreciate both Satish, Min, and in fact, Vishal also for being awake so late in the night. So thank you all of you. Over to you, Vishal.
Absolutely, yeah. But thank you, Min, again. Yeah. So last but not the least, we have our final speaker today, Dr. Anu Uppal. So thank you, Dr. Uppal, coming and serving on this speaker panel. Would like to introduce Dr. Uppal briefly. She's currently a senior scientific manager at the USP India. In her role, she collaborates with a variety of scientific experts and stakeholders to support the USP's mission on advancing quality medicines in biologics. She is also a faculty for the USP education programs and regularly conducts webinars as well as educational programs. She's an expert in mass spectrometry, more than 15 years of experience in product characterization, impurity profiling, glycan analysis, etc. Prior to joining USP, she was the lead mass spectrometry lead on in SciEx, working on monoclonal antibodies and other biotherapeutics, and was very much instrumental in setting up the basic and advanced mass spectrometry-based workflows for glycan analysis and other complexities associated with biotherapeutic modalities, right? So she has also has an expertise in analysis of peptide, metabolites, lipids, biomarkers, and discovery verification workflows. So, and she also has more than 30 research papers, peer-reviewed articles, and also has given many national and international conference presentations. So with that, I would like to again thank Dr. Uppal, and looking forward to your exciting talk about use of USP standards in characterization as well as development of biotherapeutics.
Thank you, Vishal, for the nice introduction. It's my pleasure to be here, thanks to Deepak, and talk about some of the latest developments, what USP is doing in terms of monoclonal antibodies. So with that, I'll move forward. So the first question is, what do you mean by quality? How do you define quality? Is it like if you compare a luxurious car which has fine leather seats, the top line performance, is it considered a good quality product? Or for example, if you compare a compact car which is hassle free, no repair is needed, no engine problem, so is that considered a good quality? So basically what I'm trying to say here is that quality is the mindset. So where you can compare a product with other product in that category. So for example, let me give you another example. A simple plastic is called, of better quality if it is durable, if it is microwavable. So that microwavable characteristic, its attribute, makes it a better quality as compared to others. So it's more about that how you demonstrate your product to be a better quality in terms of its attributes, its characteristics. So in terms of manufacturing, when we talk about, there are multiple considerations. Say you need to demonstrate that your product is safe, pure, potent, effective, and stable. So likewise, the attributes which are needed for this product are met with a particular level of standards. Maintain product consistency. So that means throughout your manufacturing process, throughout the product life cycle, you are showing a consistency. So for example, if it is, like its glycan profile, it's showing a consistent glycan profile throughout the process. Product testing and characterization. So you need to show with multiple test procedures and characterization, ensure that its specific criteria are met, and the attributes what are there are meeting the specifications. Implementing CGMP principles. Again, in terms of manufacturing, it is really important that you need to follow the manufacturing practices. So involve quality, build that quality right from the beginning. So the early development, the better understanding you develop of your product, and you will reap the benefits in the later stages of the product. Quality by design, it's really important to implement to develop a better understanding of your product, which can be utilized later into the late stages and into the marketing and characterization, commercialized stages like post-marketing approvals. So here, in terms of monoclonal antibodies, when we define, it's the product quality attributes or the critical quality attributes which are important to demonstrate its safety and efficacy. So as per Q6B ICH guidelines, these critical quality attributes need to be monitored and controlled throughout the process. You need to develop the analytical considerations, the analytical test, which target these CQAs, and it gives you a thorough understanding of the product profile. But when you do all these analytical tests, there is always uncertainty that are there any hidden things? How would you ensure that the procedures what you are using is taking care of all the CQAs in there? Their impact on the safety and efficacy, are you able to identify all those CQAs? Are there not any missing or hidden things? So it's really important that you understand your product very well and make sure the analytical methods what you are developing are fit for purpose. So I'm sure you might be knowing that monoclonal antibodies, being the large complex molecules, very heterogeneous, multiple tests and assays are there. Multiple critical quality attributes need to be monitored, whether it is at primary structure level where you need to identify the sequence, the PTMs, deaminations, oxidations, or is it the size or charge heterogeneity, the acidic and basic variants. So again, a lot of analytical considerations need to be taken care of to ensure that you are controlling these acidic and basic variants. Then the potency assays, the functional assays, it could be a Fab-related activity or FC-related activity. Then it's a glycan composition, higher order structure analysis. So basically, what you need to show is the totality of evidence. So if it's a new, it's a biologics, you need to make sure that you are taking care of all these things. If you are doing the biosimilar analysis, you need to show the similarity assessment. Analytical similarity assessment and totality of evidence that all these CQAs are perfectly aligned and are well consistent with the innovator molecule. So what are the basic challenges faced? So here is an example. Here you can clearly show, just give me a second. Yeah, so here you can see that, with time, how the acidic and basic variants change. It could be related due to the changes in manufacturing process, storage, transport conditions, could be anything. And even the glycoside profile, it can change over the period. So, so then the question arises, how your methods, how would you ensure that, these changes are happening because of the method artifacts? So any changes which you are seeing in your analytical methods, whether it is because of the product attribute related over the period which is happening, or is it happening because of the method artifacts? So it is really important that you differentiate these two features. So again, focusing on your analytical methods, developing your suitable system suitability standards, so optimizing your analytical method performance. So then making sure that whatever changes you are seeing over a period of time, is it not related to your maybe chromatography or column changes, or it's because of the storage condition or the temperature conditions. So it's really important to differentiate these. The other challenge is that how to ensure that the system suitability, what you have developed for your methods, is the right one. So here is an example where the particular system suitability standard was checked over a period, like day-to-day variation. So you can see a lot of variations are there over the period. So then again, how would you ensure that you're differentiating product versus method-related factors and your system suitability criteria is well defined over the different stages of your life cycle of your product. So whether it is in clinical stage, it is in commercial stage, or the early. So it is really important defining a well system suitability criteria. This is just to again highlight that multiple techniques are needed to ensure the to meet the to identify the CQAs, to follow the ICH Q6P category. So in terms of establishing the identity, purity, and stability of your product, you need to ensure that multiple tests. So and for each quality attribute, there will be orthogonal techniques needed to get the proper confirmation. So I'll be talking about some of these now in my next few slides. Here is an example, aggregate analysis using size exclusion. Sorry. So here, how would you ensure your method performance? So here is an example where you can easily differentiate between the high molecular species and the low molecular species. And when you refer to the USP chapter 1 to 9, it gives you the criteria to be considered for defining the system suitability. So let me just give you the idea that in USP, there are two different types of chapters. And these are like specifically numbered. The chapters which are above 1,000, they are more informational guidances chapter. For example, 1132, which is for host cell protein characterization. So it summarizes nicely the best practices or industry guidances, what needs to be followed to develop your SCP assays. And like here, the other chapters which are less than 1,000 are the chapter which might have some specifications defined for particular methods. For example, chapter 1 to 9, which is for monoclonal antibodies, and it talks about different methodologies, like one is the sex HPLC, and then how to really ensure that the system suitability criteria, what you are defining, will take care of your method performance. So optimizing your method performance and ensuring the consistent reproducibility. So basically you can, in this case, the system suitability, you can define by showing the chromatographic similarity. By running this standard, you can say that, okay, these conditions are met, like percentage peak area, main peak is this much percentage, and high molecular weight is between the 0.4 to 0.6%. So it actually tells you that your method performance is optimal. Likewise, running the CESDS under reduced conditions. So in order to look into some of these non-glycosylated heavy chain moieties, you can run this, and then again, some system suitability criteria are defined to assess your method performance. Another example here is the under non-reducing conditions. So looking into the product-related impurities, the degradant profiles, like heavy chain, heavy light and heavy chain combinations. So different combinations, truncated forms. By running this, again, you can ensure the method performance by looking into the resolution of these peaks, by that it should not be less than the 1.3. And then also looking into the relative peak areas. So, and again, it's like multiple standards are available where, depending upon your molecule, you can choose these standards which is more fit for your purpose and which is closest to your molecule. So idea here is that when you are running your sample, you should have that external calibrant, external standard, which helps you in optimizing your method performance. So this way you can ensure that your capillary is working as per the expectation. Charge variance, another important aspects. So here, generally, capillary electrophoresis, whether it's the CIF or ICIF, are used, or cation exchange for it can be a pH or a salt gradient. And then there are also certain emerging methods like capillary electrophoresis coupled with mass spectrometry or multi-attribute monitoring, which I'll be talking a little bit about in my next slides. So here is an example. So doing CIF, what are the advantages when you are looking into the different acidic and basic variants is that it's quick and easy. It tells you the relative quantitation of acidic and basic peaks. And actually it compares, you can compare batch to batch and multiple overall profiles. But there are certain limitations with that when you are running only C, it can tell you that what are the acidic and basic variants available, but it doesn't give you the identification. And then it also doesn't tell about modifications like deamidation and oxidation, at what position they are happening. And that is where having CE coupled with MS can be a highly advantageous, because it can give a lot more information in terms of identity of these acidic and basic variants. Other factor which came up, so you might be aware that USP actually works in close collaboration with industry and regulators. And we often conduct time to time open forums or roundtable discussions to understand the industry requirements. And one of the things which came up during these discussions was the qualification of chromatographic columns, especially when you are doing the cation exchange chromatography, whether it's a pH gradient or a salt gradient. If you are not qualifying your column properly, or you are not equilibrating your column properly, the profile can change every time you're running. So how to then ensure that the performance is consistent every time you are running your sample? So then there has to be certain system suitability criteria defined, some qualification parameters defined, which can help in, again, setting these optimal criteria. So that's where USP formed a working group, and where we are working on defining the system suitability or the parameters of consideration for column qualification. So some initial results are here based on the pH gradient and salt gradient, and this will be becoming official by early next year. Then little bit about the glycosylation. So we all know glycans can affect the efficacy of your protein, and it's really important that in terms of showing the similarity, you need to show the glycan profile. So whether it's at the intact level, it's at the glycopeptide level, or it's at the glycan level. Any changes in these like mannose can increase the clearance of MAP. It can enhance, it can reduce the binding affinity. Likewise, fucose, glicnac. So all these individual changes which can happen over a period of time, or any small changes in your manufacturing, like if you're changing a slight change in pH can affect that. So that's where impact on glycan profile is something which you need to understand right from the beginning, in the early development, from R&D stages to early development to up to keep monitoring up to the post-market approval. So it can affect conformational stability, folding accuracy. So that's where doing the analysis at all the levels is really important. This is taken from USP chapter 1084, which talks about the various glycoprotein analysis strategies. And like I mentioned, the chapter more than 1000 are mainly the best practices and guidances. And as you can see, this gives you a lot of different techniques done at the intact level, then at the glycan level or the glycopeptide level. So what are the best practices when you are doing, say, MS analysis or HPAC pad chromatography? So I would highly recommend you to just have a look at this chapter because it nicely covers all these different strategies of glycoprotein analysis. Here is an example, like again, relative quantitation of glycan. So understanding like, how much percentage of fucosylation is there is really important, or how much percentage of the mannose content. It's really important to understand, that so high mannose can adversely affect your product. So how do you ensure that again you are getting a consistent profile every time? So here, the standards available with USP, like there are 3 different standards: USP MAP 001, MAP 002, and MAP 003. It gives an idea on the relative percentages. So you can use these standards to again optimize your method performance. So even when you are doing the sample preparation involving kit, you need to understand your recovery, whether your enzymatic digestion worked well, then whether your LCMS chromatography is giving you the the MS parameters, what you have set is giving you the are you not using a very high temperature, would say, or a high ion spray voltage, so that you are degrading some of the glycan peptides, or whether you're, because it's a Healy chromatography or amide chromatography, how to ensure that you are identifying all the glycans and none of them is missing in there. So in there, again, you can use these standards to optimize the performance. It can serve as an external control to check the method performance. Then coming to the process-related impurities, here, then again, multiple strategies are there. So it is really important that you control these impurities. There are a number of chapters available in USP which you can refer to. So if it's related to, developing the understanding of the design of stability studies, this is the chapter which nicely talks about the overall life cycle management of developing a suitable stability testing program. Raw materials, so like Satish mentioned in his call, that polysorbates and excipient, it's really important to qualify your raw materials as well, and especially in when it comes to cell and gene therapy, the advanced therapies, it really becomes important. So there, the kind of raw material you are using, whether it's the risk 1, it's the risk 2. So there are different tiers of risk assessment there, depending upon the type of raw material used. If it is a source from, say, animals, then it falls into the tier 3 or tier 4 risk category. So then there should be a different control strategy program. So in the interest of time, I'm not going to use details. But I mean, if someone is interested into knowing the best practices and guidances for raw material testing and qualification, you can refer to these chapters. And then extractable and leachable. So another important area, how to ensure that, if you are using single-use containers, or in the downstream processing, if you have like this protein column chromatography, so how to ensure that those are not getting extracted or leached into your drug product. So that's where, again, some guidance chapters are available, which one can refer to. I mentioned earlier about the host cell proteins. So residual host cell proteins and residual DNA. Again, an important attributes to be considered from the immunogenicity risk assessment. So that's where the chapter 1132, it gives a detailed info on the host cell protein measurement, basically the immunoassay. So, ELISA is the gold standard for the HCP analysis. So this chapter covers a lot about what are the considerations when you are developing your antigen, what are the considerations for your antibody development, how to ensure that you are getting a dilution linearity. There are no HCPs, we call it a hitchhiker effect, that these proteins are not getting hooked and giving you a false positive results. So all these, another important thing is at what stages of development, what kind of kits to be used, whether it's the commercialized kits or the processor specific kits or the upstream specific kits. So what kind of methodologies you need to use at different stages of life cycle of the product. So all these details are given there. So I'm just giving you some snapshot or overview of what is covered in these chapters. Anything, if you're interested to know more, you can reach out to me. We can have a more discussion around that, or you can read the chapters. And these chapters are freely available. You can just download that from USP website. So now for HCP, I just would like to mention that LCMS is again taking as an orthogonal approach, it's coming up. It's emerging technology, and now recently FDA actually approved one application based on the HCP analysis by LCMS. So USP is working on that as well. So as a extended part of this chapter, there will be a new chapter on compiling the best practices and resources for host cell protein analysis using LCMS. Likewise, for residual DNA testing, there are two different standards to optimize, and then there is a complete guidance information available on chapter 1130 and 509. So 509 is it covers two different aspects, like what are the best practices for extraction procedure, and second is the quantitation, so qPCR detection. And one thing to mention here is that these chapters are not mandatory. So it is up to you if you want to use it, or these methods which are given in chapter 509, or you can use your own method. So you can use this as a guidance or information on the best practices. Little bit I'll talk about the emerging technologies. Like I mentioned earlier, CMS is one of the, again, emerging technology. And this can actually give you a lot of orthogonal information in terms of the charge variant analysis. So there are certain advantages of using CMS in terms of that because the separation is based on the charge, it gives you a better separation of these acidic and basic variants. And also glycans, like based on the reverse phase, if closely related glycans are there, they will elute under the same peak in the column. So here you get a better separation in CMS. So any closely related glycans can also be separated. And when it is coupled with MS, it gives you identification or sequence of that. And when you are covering it with MS and MS analysis, you can actually get more information in there about the location of the modification as well. So what we are doing, what USP is trying to do in here is again to compile the best practices of doing the CE analysis. With the help of these standards, then what is a good separation look like? How you will ensure that your CMS method is taking care of all your charge or acidic variants. So here is some initial experiment what we performed, and like you can say the mean peak. It's giving you a nice differentiation of all the glycan forms. And likewise, if you can slice it, you can get more identification even for deamidation, like at intact level, giving you the different types, identification of different types of these basic variants or acidic variants. The other technique which is emerging is multi-attribute methodology. This is actually a technique which allows you to monitor multiple attributes simultaneously. So it's, you can say it's that in one LCMS run, you are monitoring most of your attributes, whether it's your all the glycopeptides, if it is at peptide level, your oxidations, deamidations, N-terminal modifications, lysine variants, all can be monitored together. And this, how it helps, especially when you are doing a large sample or large batch analysis, and or even throughout the product development, it will actually help you in doing the more detailed assessment of your quality attributes. And this concept, I mean, you can refer to this paper where Rich Rogers from Amgen, he published this paper on LCMS-based MAM methodology. And they have already implemented MAM in QC environment, replacing 4 additional. So idea here is that, you know, if you can replace some of those traditional quality control assays which you do for your product characterization, like peptide mapping, capillary electrophoresis, and up to a certain level glycan analysis, replace that with one method. But here, I would say, like Amgen has done that, there are certain challenges, like having mass spec in QC as a big discussion topic right now, because you need to have, you know, I mean, mass spec in R&D traditionally has been successful, especially with the latest technology, high-end top platforms where they are more stable, gives you more sensitivity. But having that in QC environment is still a challenge. A lot of work is ongoing in that part. But Amgen has successfully demonstrated that. I would say it's still you know, a path ahead. A lot of things still needs to be done there. Just to summarize quickly, that multi-attribute methodology workflow, it basically has 2 steps. So one is your traditional way of doing peptide mapping. So you digest your antibody sample, you do the LCMS, MS search it and identify the sequence PTMs. And then you use this information to create the processing method to monitor the product quality attributes. So here, basically, you are using the information gathered over the period, and then based on that, you can define in certain percentages. For example, that a particular, say, oxidation is 4%. And then how you can define that, okay, the range is, say, from 2 to 6%. And then anything above that, it will be automatically flagged out. So then there are specific softwares used. And then the multi-attribute method can be based only on the MS1, because you're looking into the XIC, the extracted ion chromatograms of your peak, and looking into those percent modifications. So especially for large data sets, when you need to compare, you know, multiple batches, multiple samples over a period of time, how quickly you can identify that a particular attribute is going out of range. So it really helps in picking those ones. Then the another thing is new peak detection. So when, which is an integral part of the MAM workflow, again, it's like certain considerations are needed here. So how it helps is that when you are comparing your multiple samples, any new peak which is coming up, it could be, you know, any new modification, it could be new degradation peak, or your digestion didn't work properly, so it like over digested, any changes in pH, temperature rising to a new peak, any contamination. So it can flag out that again in, it can again then flag out that differences versus your you know, reference product and your sample. So here, but again, how to make sure that, you know, you're defining this new, defining the threshold, the mass accuracy, that what you are detecting as a new peak or as a new contaminant is actually a new contaminant or not a method artifact. So it has a lot of considerations need to be taken. So again, a lot of discussion is happening, that defining the best practices guidance, what needs to be done. And that's where we took this input, and then USP again has developed a expert panel which is working on drafting a general chapter on multi-attribute methodology, and also considering on developing some reference standards to help you with identifying some of these variabilities, and so that these can be minimized with the suitable system suitability considerations. Another new initiative which we have taken here is that we are building up an online community on multi-attribute methodology. So it's a online platform where anyone can join, and the idea here is to promote the discussions, share knowledge, connect, so across the boundaries. So anyone can join here. I mean, you can register using this link, mam.usp.org. Where we will be sharing the resources, information about upcoming events, lot of discussions on like what are the challenges which are there in implementing MAM in QC environment, what parameters needs to be considered, sample preparation protocols. So like how you can really control the sample preparation induced deamidations. So all those lot of technical and regulatory consideration discussions are ongoing in this community. So this is like recently launched in first week of October. There is another great resource which you can refer to. So it nicely maps out the different stages of product development of a monoclonal antibody, from cell line development to upstream, downstream, formulation, packaging. And it maps out the different USP chapters which are available, which you can refer to. And most of these chapters are completely like, complimentary, available on USP website. The different chapters, different techniques which are related to the antibody characterization. So I highly recommend you to visit this link, or you can even scan this QR code. And you can download this PDF as a quick reference to knowing more about the various chapters which are available with USP. So with that, thank you. Happy to take questions.
Yeah, thank you very much, Dr. Upal, for an excellent talk. It was very comprehensive view of the USP. So excellent. Thank you so much. What we'll do is we'll go straight from here to the panel discussion, and we'll take the questions there. So thanks again, and would like to felicitate you for this presentation. So once again, thank you for an excellent talk and your participation. Thank you.
Thank you.
Okay, so we can move into the panel discussions and take the questions from there.
Yeah, so meanwhile, if I can request both Satish and Meen to switch on your video so that we can put you on the display. Yeah, thank you.
Excellent. Thank you. So are there any questions? I know, Ravi, we can start with you. I know you had a couple of questions to Dr. Singh.
Yeah, Vishal. Yeah. So Dr. Singh is there?
Yes, he is.
Yeah, I'm here.
Yeah. So the second question I was about to ask is related to the analytical capability. So as you know, polysorbate analysis is really challenging. So with your experience, what would be the suitable method? So currently it's like, colorimetric method is there, and certain HPLC based method is there, and recently the CAD based method is there. So with your experience, like what would be the best suitable method which can be adapted to have lesser variability? That's the most challenging aspect, what we are facing currently.
Yeah, I'm not an expert in analytics of this by any means. I think it depends upon what you're looking for. You're looking for degradation, you're looking for total concentration. So I think you have to have multiple methods for this. There's the fluorescence micelle assay that's used for concentration. So, I mean, I cannot give you a good answer. I think you'll have to have multiple assays depending upon what you're looking for. Are you looking for concentration? Are you looking for intact polysorbate or degraded polysorbate, or you're characterizing the raw material. So all of these may require different methods.
Okay, thanks, Dr. Satish. So other question is related to interaction of the polysorbates and the proteins. So it's evident that when we put into the stability, generally we observe certain kind of degradation. We can call it as a decrease in the polysorbate concentration during the stability. So the major reason, is there any kind of an interaction of the protein versus polysorbate as a complex forms, or it's a true degradation of the polysorbate that can be measurable? So I mean, my question here is, generally a regulator ask whether because of this kind of degradation, what is the impact on the whole formulation?
Right. So as I mentioned, I mean, proteins can, depending upon your protein, there can be an interaction with the surfactant itself. If the protein has hydrophobic patches on them and so on, there may be more interaction than if it's a widely dispersed patches and a more polar protein. There have been studies done on the interaction of surfactants with proteins, and generally not very high levels, especially MABs and so on. There's not a huge binding of these surfactants to the protein, but there can be. So if you're not seeing, if you're losing surfactant during stability, it may just be degrading and your method is not picking it up, or your method is detecting the total content. So you may need an orthogonal method to see if there is a degradation product that you can pick up in that solution. Sorry, I lost track of your second part of the question.
Yeah, I think you have covered. So that's the second part. So whether it is directly related to the interaction, or it's purely a degradation. So because it's generally they ask an impact assessment. So
yeah, I mean, you would have to see if it was degraded or if it was lesser in concentration. That sort of was there in that O'Creavis review where they talked about shipping study at the end of shelf life. That was related to the fact that if surfactant is degrading, does it still have a protective effect near the end of shelf life of the product? So if you have 10% surfactant left, for example, is it still protective?
Yeah, that's correct. Absolutely. That point is up. So my last question is, one of the interesting study what you have shown is during the with and without agitation. So the aggregates level in the initial with agitation was observed to be high, and then it came kind of a stabilized one in your slide. So what is the reason? I mean to say, I just wanted to understand how to justify that. So that was.
And yeah. So I'm just looking at the numbers there. I mean, when there was no surfactant with agitation, you would get higher levels of agitation. And as you increase the surfactant level, the aggregation goes down with the agitation. But beyond a certain point, there is no more protective effect. You don't just because you add more surfactant doesn't mean you will get even less protection. So at some point you've saturated the interfaces, you're not going to get any more protection. So just because you can add more doesn't mean you should add more surfactant. There is a certain optimal level that has that provides adequate protection, and beyond that you don't need to add any more.
Yeah.
So it levels off. The protective effect levels off with concentration.
Yeah, I do agree on that part. Yeah, after certain concentration, it's no use. Like it's just an increase in that. Yeah. Thanks. Thank you very much for your valuable suggestions.
Thank you.
It's a great presentation indeed. So I have a question for Anu, but we know shall we continue, or I think.
Vishal, your line is muted.
I'm sorry. That's a perpetual problem, right? Yeah, no, go ahead, Ravi, please. Yeah, go ahead with your question.
Yeah, so the other question is for Anu. So one of the challenging aspects, like you are generating a system suitability for maps. So the interesting part is, so there is always kind of a degradation. There is always kind of a degradation, and sorry, there is always kind of a degradation. So in the system suitability, how do we put a shelf life kind of a thing? This is really challenging, because as the ages happens with the our any protein, your degradation contents will go up, like any of that, like aggregates or a charge variants. So what is the strategy adopted by the USP to control this, and how long one can use the system suitability?
Thank you, Ravi. So, see, that's the challenge, and that is where USP is working on developing these reference standards. There are four different types of reference standards. So one, I showed the system suitability standard, and USP takes care of, I mean, in USP, what we have, we call CSU, continued system stability use. So time to time, we keep on checking the performance of our standards, and based on that, we communicate that to the stakeholders. So any standard which is like, not suitable, not fit for use, will be intimated and will be recalled. For antibodies, again, that's the reason that under different conditions, you need to, I mean, these representative chromatograms are given in the certificate of analysis. So you can always refer to that and ensure that, that's where system suitability important comes into picture. But how will you ensure that, the standard, the chromatogram which are given in the COA is the right one, right? How you will ensure that the performance, what you are getting is right. So that's where what we are doing is a round robin study, means there are like globally 28 labs participating for these standards. And we are evaluating different techniques, whether you use the USP provided methods or you use the in-house methods. They are open to use it using their in-house methods, different instruments, different analytes, different analysts. Then we will be compiling data. So for example, like CESDS will be done in like these 28 different labs using different machines, different person, different conditions, experiments, so and different methodologies. And that is where based on that, we will come up with the these criteria and how these standards are performing over the period. So once we compile that, I think that will be a more, you can say that information will be available in the public domain, and which can be used to assess that your system suitability, your methods in-house performance. So which probably will take care of the problem of your degradants.
Thanks, Anu. Thanks a lot. So, but in general, the shelf life or expiry period of any system suitability from the USP perspective is about 6 months or a year. So, what is the general expiry you're going to give?
It depends on product to product. So, like I mean, antibodies, we can say if they are in formulation buffer are pretty stable if stored properly, right? But like peptides, they have different shelf life. Most of these peptide standards, they are as a lyophilized standard they are coming. So and small molecules, different products have different shelf life. So it varies. And actually based on that only, this CSU, I mean, there is a whole team dedicated in USP who on regular basis keeps on checking the CSU of these standards. So if there is anything like any specific product you are talking about, so then maybe we can discuss that in detail offline.
Thanks, Anu. I think I have no further questions. Thanks.
I hope I answered your question.
Absolutely. Yeah, that absolutely. So one of the interesting aspects is what you were telling is the system suitability is generally put it into the formulation buffer. So that is, I just wanted to like understand, reiterate understanding that part, because generally if it is sometimes we won't use a kind of like excipient like a polysorbate in the initial DS level. So subsequently, when the formulation happens, then there will be kind of a dilution aspects. So I do understand that putting into the formulation buffer will always have a better stability. Thanks a lot. Yeah.
And these standards actually, I mean, like I mentioned, there are 4 different standards. So one is the lyophilized powder, which is the system suitability standard. Then the other 3 standards, which we call it MAP 001, MAP 002, and 003. These are in formulation buffer, like I said, and these are actually FDA approved biosimilars. So these are like very close to the molecule which you'll be working on. I can share later on more details on that. I mean, you can just visit USP website. You will get more information. Else I'm there to discuss in detail.
Yeah, so I think there was one more question. Kailash, you had a couple of questions, but would it be possible for you to kind of come alive and ask those questions? Because they need some clarification. So if you don't mind, if you can unmute and bring those questions up, that'll be good.
Yeah, so Kailash, your audio is enabled. So if you can just ask your questions, I mean, you should be able to speak up. Yeah.
Kailash, are you still there?
Yes. Kailash, yeah, your audio is enabled. You can speak now. Yeah.
Can you hear me now?
Yes, we can hear you clear. Yeah.
First of all, thank you very much, Satish. It was a really great.
Kailash, if you can speak a little closer to your microphone, you'll have to be a little louder because your voice is clear, but it's very feeble. I mean, very low. So if you can speak a little louder, thank you.
Now comparatively is better?
Unfortunately, no, it is still very low. Yeah.
Okay, I think then proceed. I think my question basically for the Satish, and I was looking how stability specification can help us for the, the product he was talking about, the release specification. Mostly for the ICH, what I feel give a more flexibility once you have the broader specification, especially for the, justify the product shelf life. If you stick to the release specification, might be we have to compromise the, kind of the shelf life of the product. So that's why I was thinking about how USP is talking about the, like religious specification during the, control strategy. Yeah.
So you can have, I mean, some jurisdictions allow a stability specification and a release specification can be different. I don't think all jurisdictions allow it, but some do. So you can have a tighter specification on release and justify a stability spec as long as you can justify the limit from a safety efficacy perspective. I mean, that's the general rule. I mean, I don't know, I maybe said a specific.
No, absolutely fine. It's fine, Satish. I think may it be the very specific, because being a pharma specialist, all the times we look for the higher, the more and more kind of the extended shelf life of the product to avoid the retesting.
Correct.
More benefits to the patients. But once we are restrict to the shelf life, shorten shelf life, I'm sure we have to multiple times the testing, or we have a shortened shelf life for the batches. So it's fine. It's a product specific, but I thought to just check your opinion in the, the kind of the proteins we have the product. Thank you very much.
Thank you.
Yeah, just one thing to clarify there, that USP is not trying to, specify any limits or control specifications in there. I mean, that is something you will have to, develop as per your product, as per your process. So this is just to help in developing the system suitability criteria and optimizing your method performance.
Correct, Anu. Actually, the case was, displaced by Satish. That's talk about the FDA, not the USP. Absolutely. Thank you.
Yeah.
Okay, no, this is great. So, Min, one question I have for you is in terms of the sample complexity, right? How does it work with the complex samples in your method? For instance, if you're working with mRNA, right? If you have the lipid-based formulation, how does it work? Does it interfere, or if there are challenges, how do you solve those?
So it's really depend on what type of sample you get, right? If it's already lipid nanoparticle encapsulated mRNA, or if it's actually raw material mRNA kind of analysis. And actually, Moderna just published a really nice paper is talking about the mRNA can be modified and with the lipids interaction. So that's kind of, there's many different aspects how you analyze. And for mRNA raw material, that's what I showed here today. It's already very complex, and there are many assays need to be established. But obviously, with encapsulated mRNAs, you add additional complexity with LNP, and that need to be taken in consideration in terms of sample preparation. As a first start, as always started with sample preparation with fit for purpose assay development. And for different level of analysis, I showed today for cleave the mRNA with endoribonuclease T1, and that cleave into the oligonucleotides, right? So you can get a sequence coverage and assess some of the modification locations and modify the nucleoside. But there's also assays to do nucleoside and nucleotides analysis. So different orthogonal method give you different type of information. So I think as a field, this is relatively new. Obviously, for certain company like Moderna, they have been investing in the field for a very long time. They have a lot of knowledge and experience with it. But there are also many new companies looking into that space. They are going to learn in terms of how to characterize and determine the quality attributes and the critical quality attributes, and develop a appropriate assay. And even for the lipid nanoparticle lipid analysis, that's another area. So similar as final drug product, every aspect that needed to be taken in consideration, stability, everything. It's just different molecule, but the complexity is going to be more, definitely.
Makes sense.
Yeah.
Great. So we have Deepak on. That means time to conclude, right?
No, I mean, would like to really take this opportunity to thank all the three speakers, starting from Satish, Mindu, and Anu, for the wonderful talks you gave and a very interactive session. So especially Satish and Mindu, I know it's almost 1 a.m. in the morning or even beyond that for you. So we really appreciate your participation, and thank you for being there. And hopefully with Moderna's vaccine drive and other companies in place, so hopefully by next year, we should be able to do our in-person meeting, and we would be more than happy to invite you to deliver a talk in person wherever we decide to have our venue next year. But looks like we'll be having an in-person meeting. So thank you, Satish. Thank you, Mindu. And thanks a lot to Anu for giving a wonderful talk. And a special thanks to Vishal, to you as well, because for you also, it's like almost 1:30 or 1 a.m. in the morning. So you've done a great job as a moderator, and thank you for being awake so late. So before I request officially Vishal to you to conclude the webinar, let me just request all the participants that I'm just launching the poll. So feel free to post your responses, and while on your screen you will see just few questions, and you just have to respond to that, submit your response to that. And during meanwhile, Vishal will give his concluding remarks. So all of you can look at your screen, and even panelists can also participate in that. So on your screen now, you should be able to see the poll. And now would request Vishal for his concluding remarks. Over to you, Vishal.
Okay, thank you, Deepak, and thanks for all the hard work you do to make this happen. And to be honest, I stayed awake because you told me that the next meeting will be in Goa, right? So, so we'll be looking to that for sure. Yeah. So no, again, thank you for the speakers. Always a pleasure, Satish, to have you and see you virtually now. But thanks to Min and Anu, very nice meeting you, and thanks for excellent presentations and answering all the questions the folks had, right? So it was really enjoyable for me personally, and I'm sure for the audience as well. So thank you again, and have a nice rest of the day or
thank you, Vishal.
Good night. Yeah.
Thank you, Vishal. Thank you, everyone.
Thank you, everyone. And would request all the remaining panelists and attendees to just hang on there. And speakers can sign off because it's already being late for you, Satish and Mindu, and even Vishal. So we'll continue with our remaining Kindle draw, and then followed by, would request all the poster presenters who are present there in the today's call to just hang on there. We'll have a quick 5 to 10 minutes of discussion, and then we will end. So once again, thank you, Satish, Mindu, and Anu, and Vishal, and thank you to all the panelists, and special thanks to all the attendees for being so interactive and active participants. So thank you all.
Bye-bye.
And for the participants, I think the poll is still on, so you can submit your responses. Thank you, Mindu. Yeah.
Have a good one. Bye-bye.
Bye-bye. So for all the participants, would like to request to keep submitting your responses. The poll is still on your screen. You can see we have only 10% participants have responded so far. So would request all of you to please submit your responses.